Good morning. I'm Dr. M. Nishantni, Assistant Professor, Department of Biochemistry and Bioinformatics, Dr. F.J. Janaki College of Arts and Science. Today I'm going to take a topic about homology modeling. Let's see what's homology modeling. So what is homology modeling? Homology modeling is nothing but you're going to predict the three-dimensional structure of a protein sequence using the template. So the template is nothing but the protein structure which is already present in the protein data bank. In the protein data bank, there will be experimentally determined structures. It can be X-ray, NMR or electron microscopy structures. With the help of that structures, you are going to predict the structure for your protein sequence. That is nothing but the homology modeling or comparative modeling or 3D modeling. So if the similarity between the target and the template sequence is high, then we can get the good model. So now we will see the criteria for the homology modeling. So if the sequence identity is 35% to 70%, then we can proceed for the homology modeling. If the sequence identity is less than 30%, then we can go for threading method. If the sequence identity is less than 25%, we can go for ab initio prediction. So this is the overall workflow of the homology modeling. You are having your target sequence, then you are going to identify the template from the PDB. Okay, so this PD from the PDB. So how you are going to identify? Just you are going to paste the target sequence in the blast and then select the search against column as PDB. So if this blast will give number of hits. Among the hits, you have to select the template which is suitable for your target sequence. Then after the alignment, next you are going for the alignment. That is nothing but the target and the template sequence. You are going to align the sequence and then check whether the particular sequence is having most similarity with your template. Then if that similarity is found, okay, then you can proceed for your model building. That is model building. So after building the model, you have to refine your structure and check whether it is a good model or bad model. So if when you're evaluating using the Ramachandran plot, if you're getting a good model, then you can proceed for your next studies. Then if you are not finding any good model, you have to go for the iteration method. That is nothing but you're going to select the template which is suitable for your target sequence. In this homology modeling, there are 9 steps are there for homology modeling. First step is nothing but the template deduction. Okay, template deduction. Template deduction is nothing but how we are going to select the template from the blast hits. Okay, so first one, template selection is not nothing but you are going to select the template from the blast. So I told you how to select. Okay, so based upon, so see here the sequences are there and then PDB ID was there for your target sequence. PDB ID was there for your template sequence. So here you have to concentrate on identity. See here it is only 39%, right? So the gaps are more, 9% gaps are there. So you should not take this template for your study. So you should not take this type of templates for your study because the query coverage is also not full. Okay, so when you are taking the template, you have to take into the point first the query coverage. Okay, how much your query is covered to the template. So if your sequences, for example, it is 200, 1 to 200 sequences will be covered when you are taking the template. Okay, so next thing, gap should be 0 to 1%, not more than that. So when you are taking that particular template, then you will get a good model for your structure, for your protein sequence. The next step is the alignment. So once you got the template for your target sequence, you have to align the two sequences to get the better template, to get the better target sequences. So after running, you will be getting the template for your target sequences. After that, what you have to do, you have to align both sequences. After aligning that sequences, you have to see the gaps and then insertion and deletions. Okay, now you will be getting the alignment like this. Okay, after that, so here for better prediction, so for better target template predictions, template target and template predictions, you are giving some gaps to the sequences. Next one, optimize the template. Optimize this template is how far it is similar to your target sequence. Alignment option. So exchange side chains. Exchange side chains is nothing but you're going to how you are going to predict the side chains for your particular protein sequence. That is nothing but how you are going to predict the model for you. That means the backbone coordinates of that particular sequences will be copied, and then the side chains will be aligned. Okay, the side chains will be altered. That is called as rotamer. So you have to determine the best rotamer for your particular protein sequence. Rotamer is nothing but the particular amino acid sequences will have atoms. That amino acid atoms should not get clash with the other amino acid atoms. So if there is a clash between the two atoms, then there will be a steric, that particular thing it is called as steric clashes. Okay, so we have to check whether there is any steric clashes between the two atoms. So for that you have to select the best rotamer for that particular amino acid atoms. So next one is the insertion and deletions in your sequence. How you are going to check? So insertions are impossible. So when your target sequence is having insertions in your sequence, that is very impossible to predict the structure for that particular sequence. So deletions is nothing but if there is a deletion in your target sequence, then you can align your target and template sequence by introducing some gaps to get the better structure. Next, after that you have built the model, and then you have to optimize your model. So after optimization is nothing but you're going to minimize your energy for your uh, model like this. Okay, after optimization, then you're going to validate your protein. Validation is nothing but you're going to use the Ramachandran plot. In the Ramachandran plot, the residues which is present in the most favored regions are considered as the psi and phi angles like that. So that is in the red page, red column, whereas residues in the additional allowed regions, that will be in the yellow, and then the residues which is generously allowed regions will be in the sandal. Okay, so the amino acids in the disallowed regions are considered as the amino acids which is in the white region. Okay, so you have to take care of this amino acids which is in the disallowed regions. Okay, so when you are minimizing this, when you are minimizing your protein, that time what happens means that particular protein is energy is minimized, and the particular amino acid position is also changed. Then that particular amino acids will come into the allowed regions. Okay, so you have to check whether the particular amino acids, and then the amino acids, this amino acids will lead to bad model. So again, if it is bad model, again you have to check for the template. If the template is not good, if the template is not good, you will not get a good model for your structure. Okay, so the first thing, I'll just summarize the things. The first step is that you are going to select the template. Okay, select the template from the protein data bank. So once you have selected the protein template from the protein data bank, so what you have to do, you have to align the structure, align the sequence and the structure with that. So after aligning, okay, you have to check for the insertion, deletions and gaps in your sequence. So these are the three main points that you have to take care when you are aligning the target and template sequence. Once that is aligned, after that what happens means you can go for the model building. Okay, model building is nothing but you're going to copy the 3D backbone of your template to the target sequence. So backbone is one only copied. After that you are going to model the side chain. Side chain is nothing but the R groups, which is a functional group of one particular amino acid. Backbone is nothing but the peptide bonds. Okay, so you're going to model a R group for your amino acids. Okay, so amino acid side chains. So you are going to build the side chain for your target sequence. Once you have modeled the side chains, then what happens? You have to go for the optimization process. Optimization process is nothing but you're going to check the quality of your model. So once you have checked the quality of your model, again, what is the thing? So what is the thing that is creating the bad model? Okay, you have to check, and then you have to again optimize the particular protein, and then you will get a good model. With the help of this Ramachandran plot, we can analyze the amino acids which is present in the disallowed regions. Okay, so thank you for your session.